In 1903, the Wright brothers took their first flight after several years of experimenting and designing the first plane. They flew a whopping 12 seconds, traveling merely 36 meters. Then, in 1973, Neil Armstrong took the first steps on the moon. In the span of 70 years, humans went from flying glider planes to traveling into our solar system. In 1903, the majority of people believed that going into space would be impossible, but the impossible became possible. And those same people may have been watching when Neil Armstrong made his infamous quote, "That is one small step for man, one giant leap for mankind." This demonstrates that technology is developing so rapidly that anything is possible within a lifetime with a small step.
One such leap that has been taken is the engineering of genes. In this video, we will explain what is being done in that field of genetic engineering and the positives and negatives, and ultimately questioning how genetic engineering is necessary for the future of our society. In your body, you have about 32 trillion cells. These cells provide structure and support, facilitate growth through mitosis, and produce energy. Within your cell, you have a nucleus. In that nucleus, you have the building blocks of life and what makes you, your DNA. DNA stands for deoxyribonucleic acid, and on its own it does nothing. However, when added together in long sequences called genes, it becomes a key element in the human body. Genes are short lengths of DNA occupying a specific place on a chromosome. They contain the instructions to build proteins, which help run your body. Genes are heritable, meaning they are passed down from your parents and influence specific characteristics about you. If you have blue eyes, they're from your genes. If you have blonde hair, they're from your genes. Almost all physical features are predestined by your genes.
Because DNA has such a huge impact on not just your physical features, but also the food we eat, your lifespan, and inheritable genes, finding a way to edit DNA has been researched for decades. A significantly cheaper way of editing genes has been discovered recently, and this method is called CRISPR. CRISPR-Cas9 is a revolutionary way of engineering genomes. The process that CRISPR originated from is derived from the process of the body's immune system. In addition to your DNA, your body has spacer DNA. Spacer DNA separates your body's normal DNA, and what is special about it is that each spacer DNA is unique. Each spacer DNA was found to closely match viral and bacteriophage DNA. This will be important for later. Equally important, your body has a sequence of genes called Cas genes. Cas genes create helicases proteins, those that unwind DNA, and nucleases proteins, those that cut DNA. Both spacer genes and Cas genes are critically in the way the human body fight illnesses, such as the common cold or influenza. From the body's immune system, scientists were able to create the CRISPR-Cas9 system. This system works by taking a Cas protein, adding a sequence of RNA they want to edit, then create a guard RNA, which essentially holds the CRISPR RNA in place, or the RNA they want to edit. Once in place, the Cas9 protein will go to the area that is wanted to be edited, then unwind the DNA and cut it. Once cut, the host RNA can be put in the place where the DNA was cut and added, where it will fix itself. What's most important about this system is that it works on living organisms and it can cut DNA in multiple places.
Gene editing can have many beneficiaries. One example is the eradication of hereditary diseases. Huntington's disease, cystic fibrosis, hemophilia, along with many other life-altering diseases, could be a thing of the past by editing a few sequences of DNA. In addition to eradicating hereditary diseases, treatments, and retroviruses, viruses that hide in your DNA could be available for all. In 2015, scientists used CRISPR to cut the HIV virus out of living cells from patients during a trial testing, proving that it was possible. Gene editing is also used in GMO foods. Genetically modified foods can help preserve the food to last longer, so we reduce the amount of food that is wasted. Genetic engineering can also end the genetic lottery. This can mean that everybody can live more equally to decrease the amount of handicapable people. Genetic engineering can also teach scientists how to kill certain viruses and diseases, since gene editing can help us learn how viruses act and behave, which make antibiotics less costly. Gene editing could also arrest or delay aging. This has already been achieved in mice. Gene editing could offer humans lifespans twice of what it is now, perhaps to hundreds of years without loss of memory, frailty, and impotence. Of course, editing the DNA structure of any organism is going to raise a handful of ethical questions and opinions about the downside. One of which is that we could make a mistake when replacing the genetic sequence and accidentally replace the wrong strand of DNA, mistakenly creating unforeseen consequences to the organism when it matures. What would we do when this organism decides to reproduce? Similar scenario occurred in the novel Frankenstein, when the monster asked Victor Frankenstein to create a female embodiment to accompany the monster to end his suffering of loneliness. Victor agrees to create the female monster, but when he was in the process of it, he realizes that these monsters could reproduce and create an entire new species of monsters, permanently affecting the human society. This is when Victor decides to end the creation of the female monster and destroys all of his work to prevent it from ever happening. Other questions surrounding the ethicality of genome editing are: what if genome therapies are too expensive and only wealthy people can afford them? This could worsen the existing health inequalities between the rich and poor, making it even more difficult for the people in the lower class to sustain themselves. What if people use genome editing for traits unrelated to health, such as athletic ability, intelligence, or height? This would create what is known as a designer baby, which could discredit the work of natural humans, since natural humans will never be able to compete with designer babies in an event such as the Olympics or when they apply for a job. The job recruiters are always going to pick the genetically enhanced over the natural humans. This is demonstrated in the movie Gattaca. Vincent Freeman was a naturally born child, but his brother was genetically edited, so it was always difficult to compete with him, even though he was 2 years younger. However, this made Vincent very diligent, and he always walked with a chip on his shoulder. Even though Victor was very intelligent, he couldn't go to a private school when he was a child, and when he grew up, he could only work as a janitor, since he wasn't genetically enhanced like his peers. One final piece of literature that demonstrates the possible downsides of engineering humans is in the novel Brave New World. The book is set in dystopian society where everyone is genetically engineered to fit their predestined roles. On the surface, this society looks like a utopia. Everyone has a job, everyone takes a drug called Soma to feel good, and no one is discriminated. However, in order to have this society to work, everybody must relinquish their individuality. This system doesn't just remove the identity of all its citizens, but also creates a hierarchy in which no one can move up in position.
It is unclear what social and environmental effects gene editing will have on our society and environment. There are lots of unanswered questions upon the subject of genetically editing organisms. Although scientists are already utilizing the CRISPR-R technique upon living organisms, we have come to a conclusion that genetically editing humans to be immune to hereditary diseases or developing diseases is ethical. However, genetically editing humans as designer babies is unethical to the future of our society.